So we've got recombinant. Amplifying DNA is just making copies of DNA. Good? Making copies. Now we have the words here to know. If we say something is transgenic, that means we added genes from some other species. We crossed the species barrier, transgenic. If you add genes, and this is something that's being done more often now, some people think, oh, well, people are scared of GMOs, so we'll do a different kind of GMO. We'll take genes that are already in this animal or this plant, and we'll take them out, and we'll make multiple copies, and then put those back in, and it'll make 3 times the whatever it made. They call that cisgenics. Cisgenics is you're not crossing the species barrier, you're just taking something already there and amplifying it, adding copies of it. Anyway, vectors are ways to put things into a gene. You have plasmids, little loops of DNA that you could put into a bacteria real easily. You could put it into a virus and infect the bacteria or the plant or the animal with a virus. You could put it into a bacteria and infect a plant with it. That's really common with a plant bacteria called agrobacterium. You could have gene guns, like we're in our movie. You saw the gene gun they made at Cornell? You saw the really pathetic, we'll shoot the chili with a shotgun. Yeah. Anyway, so if we amplify DNA, we're going to make copies of it. This is the method. We're going to cut the DNA into little pieces. We're going to isolate, find the fragment that we want. That's usually a gene. We'll talk about the method how in a little bit, but it's with a method called electrophoresis. But you'll isolate and find the gene you want, and actually it'll be electrophoresis plus blobbing. But anyway, and then you will take that gene out and you make many, many, many copies of it. You look puzzled, Lisa. Did I leave something out?
No.
All right, good. All right. There are 2 basic methods to amplify things. You could stick it into a bacteria and grow lots of bacteria, or you could use polymerase chain reaction, which we have a lovely song about. Okay? But it'll be awesome. So polymerase chain reaction is, I might as well get this out of the way now. It depends on a special form of polymerase that we found, not that we made. Remember, like everything else, I said that's one of my big themes here, is that most of the things we need for biotech we found, and then we're like, oh, we can use that. We found plasmids. We found agrobacterium that infects plants. And we found restriction enzymes. We found DNA ligase. We find all these things and then we use them for something else. In the same way, we found a special high temperature polymerase, and it's what made, we found it in bacteria that live in hot springs. And that's the thing that made it possible to do PCR. Anyway, so if you're going to do it with bacteria, you put DNA into the bacteria, you get the bacteria to grow, and poof, you got more DNA, right? If you do it with PCR, you make copies. It's like photocopying kind of. So here's DNA cloning. If we say DNA cloning, it's another way of saying making copies. In this method, the DNA cloning is making copies with bacteria. So you have a recombinant DNA plasmid, has the gene you care about, and you grow lots of copies of the bacteria. Let them go and divide. Now you have 1, 2, 3, 4, 5. Except in real life, it'd be like a billion. But you get my point, right? All right. This is used for things like studying genetic disorders. This is used for things where you have some time. It's not used for solving a crime scene. Solving a crime scene you need to do faster. You're going to do with PCR. Thermocyclers is another name for a PCR machine. But let's go with the amplifying DNA here. You're going to take a plasmid from a bacteria and cut it with a restriction enzyme. You're going to take human DNA and cut it with a restriction enzyme. You take out the gene that you care about from the human, and you put it into the plasmid. They join together because you use the same restriction enzyme. If you don't use the same restriction enzyme for both, they won't have the same sticky ends. If they have the same sticky ends, they hold on to each other with hydrogen bonds. Then it makes it real easy for the DNA ligase to fill the space. Good? So it has to be the same restriction enzyme. Yes, that is a test question. You got to know that. It has to be the same restriction enzyme. Anyway, you put that plasmid into this bacteria and you let it divide, and boom, you got a whole bunch. And so this broth full of bacteria is now making whatever it's making copies of the DNA, but it's also making whatever the DNA says to make. That's the method we use to make insulin for people who are diabetic. That's the method we use for blood clotting proteins for hemophilics. That's what we use for clot dissolving proteins for stroke victims. All of these are proteins that are made by bacteria in vats. We gave them human genes, and the bacteria make human proteins. Works pretty dang well. And this is not one of those, oh, well, it's done in a small amount. The majority of this that you get in the hospital, these things, come from bacteria that have been modified. Good? Yes?
You use the same restriction enzymes to keep the same sticky end so they all match up?
Yes. Yeah, let's back up to the picture of restriction enzymes. So right here, if you don't, say we use these two restriction enzymes, something cut with this is not going to stick very well to something cut with this.
Got it.
So you have to use the same enzyme to cut both. And there are lots of different enzymes. And if you remember, the thing that I said was that we also now have these ultra-specific restriction enzymes that we call the zinc finger nucleases. So think of restriction enzyme as scissors, think of zinc finger nucleases as smart scissors. Think of DNA ligase as paste to put the things we cut back together. And if you cut things with the same restriction enzymes, they have matching sticky ends and they can go together. And we've got that. All right. You could also use these modified bacteria as a vector to transfer the genes. Like I said, it's real common in plant research to use a plant bacteria that infects plants. It's called agrobacterium. And it's really common to put the gene you want into agrobacterium and then infect plants that way, instead of shooting them with a gene gun. So. All right. Here are some of the things you could do. You might recognize this from an earlier slide. But your DNA cloning, you're making lots and lots of copies. Know the word DNA cloning, by the way. Since it had its own slide, you probably knew that. But anyway. So DNA cloning, make lots of copies of the DNA. You could take that DNA out, put it into a virus, and infect a person with the virus to give them the gene. You could let the bacteria do their thing, harvest the proteins that they're making, bottle it up and sell it. That's how we get insulin, like I said. You could take the gene and put it into a plant, and that's how we do most of the plant stuff. Where they say genetically modified plant resistant to common herbicide, what they're talking about there is the glyphosate resistant corn or wheat or whatever, the Roundup Ready products. That's the common herbicide they're talking about is Roundup, and the real name of Roundup is glyphosate. Okay. Actually, that's not true. Glyphosate is the major ingredient of Roundup, but Roundup has a whole bunch of things in it besides pure glyphosate. There's the whole truth. Good? All right. So now we get to talk about polymerase chain reaction.
DNA to separate.
And that's really tricky because you have to heat it up enough to break the hydrogen bonds that hold the strands together, but not enough to break the sugar phosphate backbones that hold the nucleotides in each strand. Does that make sense? So you're heating it up until you break the hydrogen bonds so you can separate the strands, but not enough to break the covalent bonds in the backbone. There are a lot of vocabulary words in there that you could have, denaturing and annealing and extending. What all does that require? You do this in a buffer solution, and it has, you have to have your template DNA that you're going to read, and you have to have primers, and you have to have all the nucleotides. So how are you going to memorize all of that?
Your beautiful song.
Yes! All right. I already had somebody else this semester who thought it'd be incredibly funny to post me singing this to Vine. I don't really care. I told them they could go ahead, so whatever. For those of you who are young, this is a parody of We Are the World. All right. So PCR, polymerase chain reaction, yeah, you'll have to know what it means. is amplifying DNA outside the cells, making copies of DNA outside the cell. This works faster than growing bacteria. And you would do this for things like, say you were in a crime scene, and in the old days you use up all the drop of blood sequencing the DNA to figure out, you said, well, we know the defendant was at the crime scene. And the defendant's lawyer says, oh, but you use lab XYZ, and we don't trust lab XYZ. And you'll just have to let him go because you used up all the blood. Well, nowadays, that's not how it works. Nowadays, they recover a drop of blood from the crime scene, and you take a tiny little copy of that blood, and you put it in a machine, and you heat the DNA up, and you separate it in half, and you make a copy, and you cool it down, you heat it up, and you cool it down. Each one of those takes about 20 minutes. So it takes about 10 hours to do 30 cycles. And after that, you got a few billion copies of the DNA, and you say, okay, what lab do you want to have analyze it? And you let the defendant pick, because it doesn't really matter, because if they can't then say there's something wrong with that lab, they chose it. And if they're, oh, we don't know, well, fine, well, we're going to use this lab, but guess what? There's enough blood that we'll do it again. You pick your, because take a little tiny copy, make a billion copies of a little tiny piece, take a billion copies of that. You can figure out whose blood is at the crime scene. Or a little sample of DNA, and you figure out who's the daddy. Right? Right? Okay. So our goal with PCR is to make billions of targeted sequences of the DNA. What does this require? It requires a template, it requires high temperature polymerase, a special one that we found in Archaea from Hot Springs. It requires synthetic primers, it requires nucleotides, and really good temperature control. Guess what? All of those are in the song. I have a link to it. I'd recommend learning the lyrics. I'll probably have 1 or 2 questions about PCR on the test. And you'll know a really catchy song to annoy all your friends with while you're at it. Oh, come on. It's a win-win. Okay. So temperature control in PCR, you're trying to get it hot enough that you can break those hydrogen bonds between the strands, all those little red dotted lines, but not break the covalent bonds between the sugars and the phosphates. So you want to separate the strands. What did they call that? There was a fancy D word for that? Denatured. Yep. Yeah, I know, it's different than the way you use the word for protein, but if you think about it, it's kind of similar, because why does a protein denature when you get it hot? Because you break all the hydrogen bonds and it changes its 3D shape. So you heat up the DNA, break all the hydrogen bonds between the strands, and they separate. Good? Good? All right, so you have to not break the covalent bonds, but break the hydrogen bonds. Tricky. If you do it a bunch of times, you get a bunch of copies of the DNA. Good, right? Oh, and I even have a note that if I didn't remember yet, it's time to play the PCR song. We can play it again. All right, never mind. All right, so once you have a lot of DNA, what the heck are you going to do with it? Well, you've made a lot of copies of the DNA, so then you have to separate it. And the way you do this, you take the DNA and you put it in, you put restriction enzyme with it and cut it. And so you start with one piece of DNA that uses restriction enzymes, and it gets cut into pieces. Some of them are kind of big, some of them are kind of tiny, some of them are bigger than big, medium, tiny. Restriction enzymes cut at different places, right? So say you have 1 billion copies of DNA, and you put them in restriction enzymes. Some of them will get cut every place the restriction enzyme could cut them. Some will be cut in only 1 or 2 places. That's why you get different length fragments. Does that make sense? They're not all going to get cut the same way. So what you'll do is you'll put this cut DNA into a gel, an agarose gel. Think ultra-fine purified Jello is basically what it looks like. But it's clear to bluish. It's made out of something called agarose. And you have, when you make this Jello, you know how you can put things in a Jello mold and you pull them out and leave little holes in the Jello? That's what you're doing. They call this thing a comb, and you put it in, and the agarose sets up, and then you pull the comb out, and it leaves a row of little holes where the teeth of the comb were. Those holes you put DNA into. DNA that you've cut with restriction enzymes. Good? Then You put it in a little chamber that is full of buffer solution, and you hook up electrodes to the chamber. And electricity will flow through the water, through the buffer. Electricity will flow, and when this happens, you're going to have a negative charge in and a positive charge in. The negative charge is by the DNA in the wells, and the positive is down here. What happens when you put two negatively charged things close together? Exactly. And what happens when you put a negative and a positive near each other? Right, so if you have all of the DNA with its sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate there, and all those phosphates have a negative charge, and you put a negative charge on the chamber on this end, it's going to try to flow away. And as the DNA is moving through the gel, it'll get closer to the positive, and then it'll be attracted and it'll want to follow it. Does that make sense? Now, there's only one thing that you need to remember here. It's the same thing that we talked about before when we did chromatography. Which molecules do you think can move through the gel faster, the big ones or the little ones? The small ones. And so because of that, you're going to end up where you get this banding pattern, where you might have a couple thousand copies of this DNA fragment here, and it's a little one. And you might have a couple thousand copies of this one here, it's a big one. They all started up here in a well, right? But they don't all move at the same speed because the little ones are going to go further down the gel. They're going to go faster, right? Now, there are a few ways to mess this up. I've tried them all by accident. College is time for experimentation. I'll give you some experiments that don't work. Wow, that's moving slow. Let's turn up the current and then go to the bathroom. Wow, that's really slow. I have time to go to the student center and buy a sub sandwich. And no, there's a big long line, and you get back. In both of those cases, you've run your DNA off the end, and this is a really boring process, and you have to stay there. I'll give you one other one. Reverse the wires so your DNA goes that direction and falls off the end immediately. I've done that one too. Yeah, not good. All right. But once you're done, then you have to stain it in a way that you can see it. One way would be to use something like ethidium bromide, which we talked about that in this class before. Do you remember I said there are chemicals around labs? We have this equipment in the back of the room, by the way, in the lab. You might remember I said there was equipment. There was a chemical that the bottle doesn't say, "Oh, caution! This has been shown to possibly cause." It just says known mutagen. Caution, known mutagen. Like this will f you up. That's what it says. It's a known mutagen. All right, that's ethidium bromide. Now they've gotten away from that. They moved to a series of chemical dyes because the reason you're doing this ethidium bromide or gel red or gel blue is when this runs through, the banding pattern is not really dark enough to see real well. And so what they do then is they put a stain that sticks to it. In our labs, if we do this, we're going to use something like methylene blue, which kind of works. In a production lab in the olden days, they'd use ethidium bromide, which is great except for the whole causing you cancer, everything. Nowadays, well, it's the truth. Nowadays, they'll probably go with a gel red or a gel blue as the dye, which is supposedly less likely to cause you cancer. But in reality, I don't know, because the thing is, the gel reds, gel blues, they are less likely to attack DNA if they get across if they get to it. Less likely than ethidium bromide. But they cross the skin easier than ethidium bromide. So it's sort of still don't touch that stuff. Anyway, but let's talk about why you would use it. You would separate the DNA, you make lots of copies of DNA, then you separate it with gel electrophoresis. You have to know that. You have to know that gel electrophoresis is used for separating DNA fragments. You have to know you cut them with restriction enzymes. You could also use it, you could use these gels that you've run. One thing you do is DNA fingerprinting. So a paternity case, a forensic case. Here, the slide before was a simulated forensics case. And right here we have victim's DNA, and here we have DNA found on the shirt and DNA found on the jeans, and then we have the defendant. And if you look at, go ahead, you shook your head. What were you saying?
It's not his blood.
Exactly, it's not his blood. The banding pattern, that doesn't mean that we know that he's innocent, but it does mean that the blood does not link him to the crime scene. Because all the blood found on the victim's shirt was the victim's blood. All the blood found on the victim's jeans, victim's blood. Because the banding pattern here, we can't see too much here from the jeans, but you can see that right there. It's a solid match. And obviously the shirt, they've done it multiple times, it's the victim's blood, right? Yeah. So we don't know if the defendant did it or not. Anyway, but I can guarantee you that the defendant's lawyer will be hauling that up in court. Anyway, so when we do this, when we're doing DNA fingerprint, we're going to look at areas of DNA that we know have a lot of variation. Remember, this is important. I put in red. That means what?
It's on the test.
Yeah, note on the test. DNA fingerprinting, like a crime scene, uses gel electrophoresis. And don't go get a sandwich while you're running your gels. All right. So, it can also be used to detect DNA variation, mutations. Oh, which again was in the song lyrics, right? Right? On the chorus. So here we have DNA, this is double-stranded DNA, and we've cut it with an enzyme, a restriction enzyme, HIND3. And when we did, we got 4 different fragment lengths. So we have 4 bands. We could cut the same DNA with a different restriction enzyme, ALU1, and it gives a different banding pattern with different lengths. So what you end up here is you get 5, and they're lined up differently. It's the same starting DNA, right? But so that way you can look at, if they do this carefully enough, you can actually count individual A's and C's and G's and T's. It's amazing. Anyway, but what's really cool, look at this. Say you are looking for changes in restriction enzymes. Well, if this person doesn't have a mutation, the enzyme DDEL1 is looking to cut here, you're going to end up with 2 pieces. Do we agree? If you have a mutation in that site, that enzyme doesn't cut anymore. See it? Because we had this mutation, it doesn't match what the enzyme is looking for, and the enzyme won't cut there. So it's a way to detect mutations, which is pretty slick. Like clearly, okay, it cut normally, yeah, okay. Oh, that's weird. Yeah, they've got some weird mutation somewhere. That's literally what the banding pattern tells you. Good? All right, so what is it doing? Gel electrophoresis is sorting the fragments by size. We're using electricity to move them because the sugar phosphate backbone full of phosphates, a little bit of a negative charge. The DNA is moving through the gel. The little pieces move faster than the big pieces. We cut it all with restriction enzymes. Does this sound incredibly familiar? You've already heard this in the last 5 minutes? Good. Everybody will be comfortable with this on a test, right? Good. All right. Like I said, we can sort if we use big enough gels or enough gels and enough restriction enzymes, we can sort individual A's and C's and G's and T's. And the cool part is that now we have machines that automate it all for us. So you can literally feed them a sample of DNA, and at the end of the day, it'll let them leave them running all day. At the end of the day, they will actually spit out a sequence and tell you what that is, A's and C's and G's and T's. Awesome. Really, really awesome. All right. So let's talk about the ways that we would, remember I said our goal is like find a specific gene. Like you cut up a genome of something and you run it through the gels and gosh, now you have to find something that you want. Well, what you do, and this works really well, say you have, you know what the protein looks like that you care about, or you know what the, say you know the protein, you could make a RNA for that, right? You could, working from the protein, work backwards to make a messenger RNA, right? But you could make it out of radioactive nucleotides. Yeah, total Peter Parker stuff. Made out of radioactive nucleotides. And then you could pull this little trick. You put the DNA in the wells, you do electrophoresis, you get everything separated out. But you need to find out what little spot has what you care about. The way you do this, you do what's called a blot, and there are different variations. There's a western blot, there's a southern blot. But the whole practical upshot is that you put a big sheet of filter paper on top of that gel. Remember the gel was sitting in wet buffer solution? You put a piece of filter paper on top, a special filter paper, and on top of that you put a whole mess of paper towels. On top of that you put a big fat book, no I'm not kidding, and the pressure, it will suck the fluid through the filter paper. And some of the DNA will stick. Most of the DNA will still be in the gel, but some will stick to the filter paper. Then you peel this filter paper off, and you put it in a Ziploc bag, literally, with radioactive RNA or pieces of radioactive DNA. Either one will work. And we call these probes. And they're the exact sequence you're looking for. And remember, they'll only stick where they find the opposite, right, the other strand. And when they do that, you can then take your filter paper that has radioactive stuff on it and put it with a big sheet of photography paper like they used to have in the black and white photography lab over there. Yes, I liked that class when I took that class here. Anyway, and you put the filter paper on top of the photography paper and leave for a little while, and you come back and the x-rays coming off of this, did I mention it was radioactive? The x-rays coming off of it would expose the filter paper. It's like, oh, that's where it is. And then you'd go back to the gel and you would cut right there. Well, sorry, you would go back to the gel and you would cut right there, and you would take the piece out, and you have now captured the genes you care about. Okay, so genetic markers. Hank talked about this. We did the video. Do you remember when we did the video about the glowing rats? Why do people care about rats, glowing rats? And his response was, well, it's actually useful because you can use it as a marker sequence, but everybody else gets more excited about the glowing. And he's right, but here's the thing. You can take a plasmid, a loop of DNA, and you can put into it all the things that you need, all the things you care about. So you have this little vector, this little plasmid, but if you add a marker gene, then you know if the plasmid was picked up by the bacteria you're trying to get it into. How do you know if the bacteria was transformed? Well, it's pretty dang easy. Put a black light. The colony that glows, that's the one that has the gene you want. The colony that doesn't, that's the one that you leave it alone. So the one that glows, you come with a little knife and you cut it out of the gel, and presto, those are the bacteria that are transformed. Now put those in the vat and grow them, and they'll make the insulin or the whatever. Does that make sense? Yeah. And also then you shine a black light on, they'll glow, and that'll be cool too. So, green fluorescent protein, it is the biggie. It is the famous one. It came from a jellyfish. And we've now put it into everything. We put it into bacteria. Like I said, if you want to know if they picked it up, you hit the bacteria with the black light, they glow. You want to look at the worm C. elegans and see where it's expressing a gene you care about? Link that gene to the GFP, shine it on, so, oh, that's where it's using it. All right, so check this out. Here in Polk County, they have taken the gene from red grapes that makes them red, and they've stuck it into citrus plants, in the leaves. So instead of nice green leaves, you have purple leaves. And it is the same thing, it's a marker gene. They're doing this because they can see if some other gene they care about that they tried to introduce actually is in that plant. All right, so let's talk about DNA technology, personalized medicine, custom treatments. Did we already talk about gene editing with the zinc finger nucleases, how they took a gene out that made a surface protein on white blood cells?
Sort of.
Did I show you? Yeah, that's what I'm talking about with HIV. I showed you the video. It wasn't sort of, you got everything I got.
Okay.
Yeah, that's what I've got on it. But it's a good example. You could add genes or you could remove genes. In this case, they took out a gene that makes a protein that makes the white blood cell susceptible to to HIV, and they then put in the allele, the version of the gene that is immune because it doesn't make the protein, right? If you don't make the protein, there's nothing for the virus to grab onto. Those white blood cells are going to survive. Good? And so we can do that. Now, unfortunately, personalized medicine, personalized gene therapy, very expensive. This is not something that we can do all over the world yet. Alba the bunny. Here are genetically modified salmon. These salmon are the same age. One bulks up a lot faster than the other, right? Which one do you think salmon farmers want to grow? Yes, exactly. I don't know that this has been approved yet. They've been talking about approving it for 10 years. But salmon, actually they may have approved it already. Let's see if they have. So it has been approved in Canada. Don't know about the US. The FDA, August 3rd, so it's August 13th. They hadn't decided whether or not we could sell this in the US, but the giant salmon, fast-growing salmon can be sold in Canada. All right, but anyway, they grow faster. That's fish that grow faster. And that could be a good thing or a bad thing. Watch the movie End of the Line and you'll know why it may not be a good thing. But on the whole, I'm not too freaked out about it. Here are things that we make through biotech. We've put human genes into E. coli. You know, that same, remember I said that bacteria that comes from our backside is the white lab rat bacteria? Yeah, we've put human insulin genes into growth genes. Taxol, which is a cancer drug. Luciferase, the same one that made the tobacco plants glow. We put that in there too. Human blood clotting factors, adenosine deaminase. All of these, we make these by putting the gene into bacteria, let the bacteria grow, and we harvest the proteins. All right, DNA sequences. You could also put the DNA into E. coli or do it with PCR. You could put it into a gene machine. Gene machines are the coolest. I've only seen one once. Again, the size of a toaster, and you literally hook it up to a computer and you say, this is what I want you to make. And you give it little vials of chemicals with A's and C's and G's and T's, and it will build the DNA that you want. And at the end of the day, you have a vial full of that gene. And it doesn't have to be something that existed before. It can be something you design and create, and it'll make it. This is, like I said, a theme throughout the chapter. I want you to know that we borrowed our tools. I want you to know that we found restriction enzymes. They're used by bacteria to destroy viruses. We now make better ones, more accurate ones, synthetically, zinc finger polynucleases. But we found restriction enzymes. We didn't invent them. We found DNA ligase. Remember, it's the stuff that hooks the Okazaki fragments? Yeah. Hey, we found the high temperature polymerase, Taq polymerase. It lives in the hot springs. We found it and we use it. We found plasmids. We found agrobacteria. We didn't invent this stuff, but the minute we saw what it did in nature, we were like, hey, we could use that in the lab to do the same thing, you know? So we borrowed our tools from nature. We didn't invent them. Well, let's talk about cloning a little bit. Cloning plants is easy. We've been doing it as long as we've been growing plants. If you take cutting off a plant and you grow it, you just cloned that plant, right? Most animals, you can't do that. There are a few fun ones you can, like planaria. A few animals you can do that, but plants you can do this all the time, right? That's easy. Cloning animals is much harder. Cloning animals, reproductive cloning, we can do this now. We can remove the nucleus from an egg, and we fuse the egg without a nucleus with the cell we want to clone. And that will turn into an embryo, and we put it into the mother. I want you to know those steps. Here's the pretty picture of it. So that's Dolly the sheep. She's the first cloned sheep. And the way we did this, we wanted to make sure people knew that she was not of that mother that bore her. So the way we did it, we took a Scottish blackface egg and we sucked out the nucleus. Good? And then we took one of her, they didn't even want it to be a normal reproductive cell, so that we could say, oh, well, it only works through reproductive cell. So they didn't use something from the ovaries. They took a piece of her udder. They took a little tissue sample of an ordinary body cell, and they then fused this egg without a nucleus with the udder cell, and they zapped it with some electricity. There is a reason for that. It's not just crazy mad scientist stuff. The reason is when a sperm goes into an egg, you have what they call the fast block against panspermia. And what that means is that there's a change in the membrane potential, a little bit of electricity that says, hey, I've been fertilized, and it stops you from getting too many sperm coming in, because you don't want the egg fertilized by 12 sperm, you want it by 1. So the first one gets in, and there's an electrical change in the membrane, and that sperm is over here doing the happy touchdown dance, because I'm inside and you're on the outside, and near, you're stuck. That happens when you have a little electrical signal. So they simulate that electrical signal. They can do it chemically too. But they give it a little signal and say, hey, you've been fertilized. And the egg checks and says, well, I do have a 2N nucleus now. I must have been fertilized. And then it starts off dividing and makes an embryo. And then they stick it into an animal. And then it grows up and it raises a baby that is not related to it at all. So this is what I would have played for you guys, but we don't really have the time to play it much. So we'll play a little piece of it. This is a bull. His name is Chance. He's big and he's famous. What, you know about him?
We talked about it last class.
Oh, we did. Okay.
You just never showed it.
Okay. Well, that's Chance. If I already talked about it, I don't need. So I told you what happened, didn't I?
I think me and Lisa were the only two in there though.
Okay. All right, everybody. Here's the thing. This bull died, and the owners wanted it cloned back, and they asked Texas A&M if they would do it. And Texas A&M said, well, sure. But their reason for it was not that they really wanted to help this very rich guy who did animal performances. It was, hey, we've never tried cloning a really old animal. Let's see if it works. Well, the technique worked fine. And they got a baby bull. His name was Second Chance. Haha. But this is the cute little cowlet they made. Isn't he neat? Okay. Little baby bull. And like, oh, he's so great. And they went on about how he acted just like, all his mannerisms were the same and everything. And then it turned out he wasn't quite the same, and he attacked and gored his owner twice. The second time leaving him needing 80 stitches with his scrotum torn open and his left testicle hanging out.
Oh, that.
The thing to remember here, the owners were incredibly shocked by this. Oh my God! But the scientists were like, it's not the same animal. It's genetically identical, but that doesn't make it the same animal. It's like twins are not the same. That's okay. I've been bitten. Ow, he has teeth. Ow, ow, ow.